Assalamualaikum warahmatullahi wabarakatuh. I'm Nazifah and my friends Nurul Pasaribu, Zahrona Azijah and Annisa Zahra. We are from Group 1. In this video, me and my friends are going to explain to you about glycolysis. Glycolysis is an energy-releasing reaction that breaks down a glucose molecule consisting of 6 carbon atoms, or another monosaccharide into 2 molecules of pyruvate acid consisting of 3 carbon atoms, 2 NADH or nicotinamide adenine dinucleotide H, and 2 ATP. Glycolysis is the first step in aerobic respiration to produce energy in the form of ATP. The ATP produced in glycolysis will be used for various processes that require energy. Because ATP is an energy storage molecule, so in glycolysis there is a surplus of ATP. More ATP is produced than used in the process.
So, I will talk about the steps of glycolysis. First, we start from glucose, then glucose become glucose 6-phosphate, then become fructose 6-phosphate. Glucose become glucose 6-phosphate need ATP or energy. Also, fructose 6-phosphate and fructose 1,6-diphosphate also need energy or need ATP. And then fructose 1,6-diphosphate divided by 2 become glyceraldehyde 3-phosphate. Then glyceraldehyde 3-phosphate become 1,3-phosphoglycerol acid. Phosphoglycerol acid produce H plus and NADH. 1,3-phosphoglycerol acid also need ADP to become energy or to produce energy. Glyceraldehyde 3-phosphate or to become 1,3-phosphoglycerol acid need dehydrogenase enzyme. Also 1,3-phosphoglycerol acid become 3-phosphoglycerol acid need phosphoglycerol kinase enzyme. In this video, you can see what is NADH or NITP.
So the end result of this glycolysis is 2 pyruvic acid, 2 NADH, 2 ATP, and 2 hydrogen.
Next, I'm gonna explain you about 10 enzymes in glycolysis and their functions. The first step in glycolysis is the conversion of D-glucose into glucose 6-phosphate. The enzyme that catalyzes this reaction is hexokinase. The second reaction of glycolysis is the rearrangement of glucose 6-phosphate into fructose 6-phosphate by glucose phosphate isomerase. Phosphofructokinase with magnesium as a cofactor changes fructose 6-phosphate into fructose 1,6-bisphosphate. The enzyme aldolase splits fructose 1,6-bisphosphate into 2 sugars that are isomers of each other. These 2 sugars are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate dehydrogenates and adds an inorganic phosphate to glyceraldehyde 3-phosphate to produce 1,3-bisphosphoglycerate. Phosphoglycerate kinase transfers a phosphate group from 1,3-bisphosphoglycerate to ADP to form ATP and 3-phosphoglycerate. The enzyme phosphoglycerol mutase relocates the P from 3-phosphoglycerate from the 3rd carbon to the 2nd carbon to form 2-phosphoglycerate. The enzyme enolase removes a molecule of water from 2-phosphoglycerate to form phosphoenol pyruvic acid or PEP. And for the last step, which is 10, the enzyme pyruvate kinase transfers a P from phosphoenol pyruvate to ADP to form pyruvic acid and ATP. And that's all from us. Thank you for your attention.